Okay, I announced that's the first breakout session for infectious disease, and actually, so the main purpose of this discussion is which kind of project we should propose. It means directly which kind of disease we should. So because there are not so many people, I just briefly introduce each of you quickly, name, institution, and your disease of interest. So if you the people who do not want do not do, but if there anyone can make a brief presentation, introduction less than 30 seconds. Yeah.
Myself, Dr. Ramkumar Nema. I'm working as a scientist in ICMR setup in the National Institute for Research in Environmental Health. So I'm working on enteric viruses. I'm trying to get these viruses and want to type these viruses by using Doppler digital PCR and NGS technology and in environmental samples.
Okay, thank you. It's very classes, right? And
I am Ashish Kumar Vyas. I am working as assistant professor here in Liver Foundation. I work on placental immunology, working in hepatitis B mother to baby vertical transmission.
Thank you.
Hello, my name is Dipesh Kare. I am from All India Institute of Medical Sciences, Bhopal, and my area of interest is dengue virus and the virus-host interaction, especially.
Thank you.
I am Dr. Sungnak. I am from Budelgan Medical College, Government Medical College in Central India, and actually I am a principal investigator at the virology lab there, and I am working in chikungunya, hepatitis B virus, and as well as hepatitis B. Hepatitis B virus, and apart from that, we are also working since earlier on. We had a tuberculosis lab since 2013 itself. So we are working on mycobacterium tuberculosis and its interaction with the human microbiome and the TB immunology.
Thank you. Thank you. Yeah.
Hello, I'm Chandrika from NIBMG. My PhD was on host genetics of tuberculosis. After that, I'm a part of Genome India project, and I'm one of the lead analysts working on sequence data of 10K individuals.
Thank you. Yep.
I'm Haya Akrin, and I'm a grad student, and my topic is archaic introversion. Totally different. I'm here just to see.
Good.
I'm Soumya Basu. I'm an ophthalmologist. I work in Hyderabad, southern India. I work on tuberculosis of the eye, where we not only get microbial immune responses, but also autoimmune responses in the same anatomical space with both microbial and autoimmune.
Thank you.
Hi, I'm Bhashyavi Pandit from National Institute of Biomedical Genomics. I work on tuberculosis, the host response in tuberculosis, looking into the genomics of host as well as gene response in the host pathogen interaction. And recently, I have expanded my study towards the pathogen. There is a project pan-India sequencing the mycobacterial genome, so we are part of that. And also, recently I have started thinking in the direction of the microbiome along with the tuberculosis infection of the host.
Okay.
Hello, I am Isha Bhattacharjee from National Institute of Biomedical Genomics. I am also a grad student and I am working on the maternal and child health.
Thank you.
Hello, so myself Sungnak from NIBMG. So currently I'm working with on oral cancer actually. So I'm here because maybe in the future we are planning, our lab is planning to work on the bacterial infestation within the oral cancer microbiome, for cancer microenvironment. So
myself Waradon from NIBMG, and I am a computer biologist and I'm working on complex diseases. Complex diseases, type 2 diabetes and CVD.
Hi, I'm Veena Patil. I'm from National Institute of Immunology, faculty there.
So we study T-cell immune memory to various viral infections, and we also study overall immune response to sepsis in humans. And my grad students will introduce themselves.
Hi, I'm Shreya Sana. I'm from National Institute of Virology, and my research interest is neonatal sepsis.
Hi, I'm Kurt from the National Institute of Virology. I'm looking at CD4 and terminals.
My name is Yukinori from Okada University, and we are adaptive immune response, and we are corona and influenza specific.
Hi, I am Yukinori from National Institute of Virology, and right now I'm working on a virus-specific memory T-cell.
Hi, I'm Monica. I'm from Okada Institute of Technology in Indonesia. Our work is Yes. Okay. Our work is on Indonesian Genome Diversity Project to study association between genetic diversity and disease, such as malaria or lifestyle-related disease. Thank you.
Hello, my name is Mohan Fahro. I'm a postdoc at the St. Vincent's Institute in Melbourne, Australia. I'm in this project with Monica, and also my PI Irene. Profiling the immune cells of the Indonesian population. So we're not looking at any specific disease, but we're interested in all possible tropical diseases that exist in Indonesia, like malaria, typhoid, dengue, and so on.
I'm Irene Gallego Romero. I'm a co-leader at St. Vincent's Institute in Melbourne, in Australia. And like Monica and Hashim have said, we're interested in any sort of infectious disease and blood that we find in Indonesia, with a focus, I think, on malaria and dengue at this point in time. Hello, everyone. I'm Shejyuti. I'm a director and senior scientist at the Research Foundation in Bangladesh. My team primarily focuses on pediatric infections, so sepsis, pneumonia, meningitis, and typhoid. And I also founded and run the Genomic Center at the Child Health Research Foundation.
Hi, everyone. My name is Ryuya Erahiro. I'm a respiratory physician and a PhD candidate in Osaka University, Japan. My interest in the host immune response to especially COVID-19 and LTM. Thank you.
Hi, I'm Seitaro Nomura, Japanese cardiologist, and I gave a short talk regarding single cell analysis in the heart. I'm also interested in inflammatory cardiac disease, such as cardiac sarcoidosis and COVID-19 related myocarditis. Thank you so much.
Hi, myself Bijurika Chakraborty, and just a couple of months back, I have been awarded my PhD degree, and where the main focus of my thesis was post-transcriptome of malaria pathogenesis. And currently, I am recruited as a research scientist in NRS Medical College, where I have actually come to a real-life scenario where I can see how patients, whatever the disease may be, one of the main problem or one of the main risk factor that is caused in them is infection. So I guess this is a very apt session where I should attend. Thank you.
I'm Abhilash and I'm from IIT Delhi, and I work on the transcriptomics in the splicing, how splicing affects the lung and liver disorders. Thank you.
Hello everyone, my name is Eva. I'm from Uganda, and my interest and what I work on is really differences in vaccine responses in urban and rural population, and of course these are in the presence of pathogen malaria and to be specific. today. My name is Nitai. So what I do is basically after my retirement, I spend time with the clinicians in SSKM hospital. And what I'm now trying to do, try to understand things and whether these ideas can be translated into the specific, solving some specific clinical problem. So it's a generating idea generating process.
Okay, thank you. Okay, so yes, we have some participants from online. So from my view, the top is the Mr. Collins Moran. If you have some comments, interest in some of these, please let us know.
Thank you very much. My name is Collins Morana. I'm an infectious disease scientist from Ghana, and I'm attending online. I work on malaria infections. Thank you.
Okay, thank you. And the second people from so Paul Namuk, yes, again, yeah. Can you introduce yourself and your disease of interest?
My name is Paul Namura from Keio University, Tokyo, Japan. So I'm an ID physician and I also interested in the host genetics of primary infectious disease such as COVID-19, tuberculosis, and NTM. Very nice to meet you.
Thank you. Now we are really happy to hear your voice. Okay. Is there people? Mr. Miss Ansel Lim? If is any comments or who named Prier? Okay, maybe we can let's start some discussion. So, thank you everyone coming together. And again, so this is a breakout session, so we may have much more detailed discussion on this point. So actually, we didn't have so much time to discuss, but actually, there's several points we should decide within this meeting. So this namely short-term goals. So, namely, yes, maybe which of the infectious disease. Of course, many kinds of infectious diseases of interest, but because we need some collaborative network, we should definitely focus on some systems. So we have just made a quick survey of your disease of interest, but as long as I have heard correctly, what I heard is TB and dengue and hepatitis and NTM and some vaccine-related responses. So actually, we put so many, but I think it looks like the disease of interest somehow more centralized, and I think. Yes, the disease only occurring in one or two Asian countries is not a good target for this project. And disease occurring all over the world is also not good target. So I think in some sense, some focusing problem in the Asian countries is also fine. So this is these things. And second thing is, so which kind of technology we should use? Of course, we have already, or most of us already conducted just single cell analysis of blood, 5', but just we do more advanced technology to how to make the good, how to solve the problems. The point is sometimes tissues. So should we focus on blood or other than skin or clone or swab or somehow? So the target tissue is very important. The second point is the technical point. We can, of course, we should use RNA-seq, but we should somehow consider attack or much or more long-read or spatial transcriptome, which is strongly related to what we are doing in the tissue of the selected ones. And also, the other important point in infection is we should also need to the pathogen side, not only host side. So also strain level diversity is different. I think that even though TB, the sub-strains are different across or within Asian countries. So we also need to find a way to define the strains of the target infection. And it's also how to make more, how to see the pathology analysis? Just see looking for the infected patients, or going to see more longitudinal changes or vaccine responses. Also, we should focus more on adaptive immune response side T-cell VCR. Okay, and so this is maybe the point of the discussion we should talk, but maybe I should start some open discussion for everyone who have some your opinion into each of these 5 to 6 points. So people who have your opinion, please raise your hand and yeah, please go ahead.
Thank you. So the reason why I'm interested in the analysis of the influenza-specific T-cell response among Asian countries is that based on our preliminary results, a comparison between Japan and the Vietnamese people on spike protein specific. So there are really huge differences between Japan and Vietnam countries. That's the reason of the best. First, it's locations and the infectious disease.
Sorry.
Okay. Also, plus vaccine histories. So what I realized was there's a major differences on the baseline immunological system among different countries, and particularly in Asia, it's a very kind of interesting locations because COVID-19 and avian influenza, they're all derived from South China areas. So many Asian countries has been exposed to very similar viruses and the reagents. So by looking at the kind of heterogeneity of the antigen-specific response among these Asian countries, including Japan, Korea, and India, and Indonesia, I think that could be really, fundamental. Will give us fundamental insights about the immune system and the differences among countries.
Yeah, thank you. I fully agree with your thoughts. So maybe you say it's because each people in different countries have different background exposure, so the response to the vaccination may reflect each of the different aspects. Yeah, I fully agree. So I mean, you're just focusing on the response to the vaccination, right?
No, it's not only vaccines. It can be like infectious diseases. So like my expectation, what I'm thinking that by looking at influenza T-cell response, so as a kind of from healthy volunteers, I think there is a huge variations among countries. So we don't need to get infected patient sample. We can get the healthy volunteer samples, for example, blood samples, and we can look at the other type of the T-cell responses.
I mean, you get sample and do some experiments on stimulation or vaccination, right?
That's right. So we need to somehow the T-cells.
I see. So yes, so maybe so we haven't already discussed on the which disease, but trying to figure out the difference of immune response. I think Asian diversity is really good to have. Thank you. Yeah. Good. Thank you for your good user suggestions. Yes. So, is there any comments or discussions on this point? Maybe I have a question. So we just talked about some, for example, vaccination stuff. So I'm not sure, but is every country in Asia for example, have vaccination of influenza annually or not? Or, for example, in Japan, we always have annual vaccination of influenza, but I'm not sure how it's going in other Asian countries. How is the environment in your country?
It's recommended, but it's not required. So it's another area, right? So like in one area, so from what I gather, it's more like Asian immune diversity at last plus, right? Like baseline in healthy, and then extend toward antigen-specific responses. But then, so that's one idea, right? But any other ideas that like anyone in the audience would have any I don't know, like opinions whether we could kind of coming together and then propose something as a flagship project.
No. Please go ahead.
I think there is scope for both directions in the same proposal, right? In one proposal, you characterize baseline diversity using standardized stimulations in a laboratory setting, which is something that we're doing also in Papua New Guinea. And then you can also have, let's look though at like pathogens and infections in not a controlled environment or opportunistically. So some of our samples that we collect for AIDA, we know have malaria. And so we're interested in looking at those people who are not presenting necessarily as ill, but they have a detectable load, and understanding all of those interactions, I think, is perhaps more valuable. There is space for that in a broader project that focuses in infectious disease in general.
So should there be any guidelines for collecting so like healthy is easier, right? Healthy is just healthy, maybe like, but then like infected, then the like confounding factors, like like when did you get infected, and then like is it I don't know, like early, is it late, convalescent, so that so many more parameters. So what do you think like we should do in terms of like controlling for these parameters?
I actually want to push back. I don't think defining healthy is easy at all. Very complicated, and it varies a lot from site to site. But I think, yes, you need some sort of standardization, and you can have different cohorts, right? You can have just like a natural cohort where you say, we sample people in this setting. Some of them had X, Y, Z. We detected afterwards, and some of them were free of pathogens that we looked at, and that's what we saw. And then in another setting, you can have targeted recruitments and go to the hospital with X, Allison, whatever. But you can do all of them. Again, those distinctions, those different cohorts, maybe come together as a singular big project.
So should there be any specified or list of pathogen we should look at? Like I could see like healthy, right? Like because we are working on like asymptomatic infection as well, right? So like unless we actually detect like targets, then we would just call the person healthy, right? So that's complication that makes like infectious disease kind of much more complicated, like in addition to the healthy alone. So from that area, like simulation from the healthy and then multiple cohorts, probably like just collecting like the metadata, like what you do with the information related to specific cohorts.
I think in infectious area, everyone with biology or immunology, we should have a database with specific protocols, because all the lab have some different the procedure of doing the same thing, and they're showing the data or submitting in different portals. So if we have a specific catalogs for like stimulation assay or in vitro analysis or something, then it may be useful for.
Yeah, I think so. So one of the bottleneck of this type of international collaboration infection is how do we handle the samples from infected people, or how to unify the stimulation stuff. So, for example, when we did AIDA, so AIDA consortium, the single cell equity of the healthy control, I think the story was relatively easy. So each country has collect just EVM share healthy people and do the tags and do the analysis. But infection points, sometimes the status of the infected people or sample is different in each country. And I think stimulation stuff, those things is sometimes difficult to do the same things together in different institutes. But it is also hard to just send living cells, right? So maybe because this is a kind of international collaboration, we should pave the way so each institute can do the same things. So yes, this is one of the bottlenecks. And also, what I have forgot to say is, I think that some midterm goal is Pan-Asian network of product. It means that maybe we initially start focusing on some major disease of interest, but I imagine, or within some years, not so big, but small scale of regional pandemic may occur in Japan, or in Asia. I think I want to have a scheme which can quickly assess such event in the Pan-Asian network. So in this sense, we need to make network also support of the IRB. So I mean, every country can get together to collect the sample, and then once some new infection disease occurs, let's just collect the sample in the same way, and then we can do the research quickly. So anyway, we need to make, even though we are focusing on the simple disease, we need to make networking. Everywhere we can share sample and data. So we need to think which kind of procedure everyone can feasible to join. So this is an important point. So yes, people who have some interest in some stress disease or sample, please let us know what is the best way to do. But yeah,
can I add another point for discussion? Like maybe asking for your opinion or anyone's opinion as well. So as someone who work on infectious disease using like single cell transcriptomics and like TCR, BCR, what I find to be kind of a hustle for me is that like in other areas when people work on like human cell atlas, there are cool tools coming up like every now and then, right? But then tools that are supposed to be used in terms of looking at infectious diseases are quite lacking, and a lot of time like we have to jump in and try to find things yourself. So one of the example was like what Jinmao like presented earlier, right? Like we can just look at viral RNA, right, in just our 10 example. So in theory, right, as long as we have reference that are for like microbes or even pathogen, so could we develop something that a list of common pathogens that we commonly find in human? Like could that be like a standard being added to maybe like Cell Ranger or the like analysis pipeline? Is that possible, right? So that's one thing that is lacking. And like other things like like information related to TCR, BCR, right? People talk about coring like like cell type annotation and all the data. And like we all know, or some of us know, that if you want to look at antigen-specific sequences, we have to look at like VDJ-DD, right? Like a different database, even though some of those are already single-cell data. So is it possible that like we could incorporate VDJ data, like antigen specificity, being added to all the like atlases that like people talk about already, so that when people who are interested in infectious disease want to mine and try to find like like initial understanding of anything, right? Like it can be readily available. So so that's something like the struggle, like myself and I kind of want to, if we have some effort as a community, maybe we would have a better momentum for pushing for like getting this tool like developed faster. So I think we got a lot of credit from like COVID-19, right? But then, yeah, we still need that. Yeah.
I wanted to ask, the list that you have is missing fungal pathogens. Is there any reason why fungal pathogens are missing from the list?
What? Pathogen?
Fungal? Fungi? Yeast?
Fungus. Yes, definitely. Yes. So for the pathogen side, yes, it's not only restricted to viruses. So actually, or virus are typical bacteria. So fungus is disease with fungus is also of interest. And yes, any TME or TB is not a bacteria, right? Different types. So yes, not restricted. Yeah. So for example, fungus. Which kind of disease you are focusing on as infectious disease targets? Fungus infection.
Candida albicans, Candida auris.
Yeah, Candida for.
Another question that I wanted to ask is if you have any guidelines for biosafety handling of such samples.
Yes, it's difficult thing. So I think actually sending the biosample to abroad sometimes hard. So each institute may do the analysis, make the scheme do the analysis sample by themselves, and they share the data is the quickest way to use. But yeah, it depends on this.
I'm sorry to interject, but I just need to make an announcement. This session is supposed to finish at 4:40, but we have back-to-back sessions, so you don't have to finish this session right away. The announcement that I want to make is that if anybody wants to swap rooms, feel free to do so. Okay, this is a continuous session that will continue, but if you want to move from one room to another, feel free to do so. Thank you.
Go to the so that's something that like we kind of included in the challenges slide already, right? Like as you can see, like everyone works on like different diseases, and then the examples that we gave today, it might not include diseases that like you might be interested in. So like that's one thing, right? That we like to gather your opinions from this discussion, right? Like like how should we like take your like feedback and then like like make this potential flagship project to kind of like like satisfactory for or as a consensus for everyone.
Yeah, so this is the breakout session. So any kinds of discussion from audience is welcome. So please raise your hand. Yeah.
Yeah. I think I'm following. So I think right now what is really needed in Asia, specifically countries that are a lower middle income, is basically capacity, right? Capacity strengthening both for doing the work themselves or ourselves in the country, with library prep, sequencing, single cell capture, and then the bioinformatics. I think something that I would disagree with a little bit is sample sharing is becoming harder every day. And sample sharing is also not sustainable, right? The countries who did the best work during COVID, at least the initial year and a half, are the countries who already had capacity within country. Because during COVID, not only was it hard to send samples out, get our act together, there were no flights to even ship samples. And of course now with the WHO Nagoya protocol, etc., it's really difficult, if not impossible, to ship biological samples. So if we want to think about long-term sustainability of work and really contribute to Human Cell Atlas, it has to be, I think, data that's generated within country, data that's analyzed by people in country who understand the context. So I think if I were to really get very excited about a flagship project, it would be something that comes up again and again, rather than you also brought it up, is shared protocols. We all have different interests here. We have cardiologists, people interested in fungi, people interested in dengue. We have our own interests, we have our different problems, we have our different networks, own networks in the country. So I think rather than, trying to specify a specific disease or a pathogen, perhaps something we could do is a specific sample, right? Like, share protocols for one type of sample that is relevant to most infectious diseases. That way we don't have to move very far away from what we currently do or what our interest is. Once we set on one or two types of samples, we can share good protocols that all the teams could use and then compare the results, even if it is different pathogen. And I really, really like that idea of having a database of pathogens that we pick up just by doing single-cell genomics, but are never recorded. Like even in the morning, Shyam was asking those questions about, we see these reads, how much of it is real, how much of it is not real. We don't know, because most of the data that we have so far has come from Northern Europe, right? Or descendants of Northern Europe. So I think the more we generate these data, and if we can really have a database of, what are pathogens that are just circulating in our blood or in our nasopharynx all the time, would really give us a good baseline to go forward. Over.
Yeah, thank you. Truly appreciate your opinions. Actually, sharing biosample, especially infection, as you heard about. So, your opinion is to make the shareable data protocol to share the protocol to the shared protocol with doing assay and making data, and then share the data. And the protocol assay is more generous. Generally applicable many types of infectious disease, or at least some specific list of the diseases, and everyone can join each disease project. I fully agree. So actually, it may be there's some practical way of doing it, and good. Oh yes, please. Yeah.
So I think even data sharing can be quite challenging, certainly at the level of raw reads. And so I think it's also worth thinking about federating and data sharing in that context where data remains in each country, and the analysis itself is what's federated to really sort of deal with these concerns about sharing and ownership and extractive practices. I think it's worth considering that, and again, focusing more, I agree very, very strongly on sampling and establishing protocols, but also on training. So something like similar to what HCA Africa does, where they have these training courses, would be, I think, like training days would be really, really valuable, I think, in this context in terms of building local expertise.
Hello.
Oh, please.
Host pathogen interaction is an important aspect in infectious disease. So we can think of not looking into the host or the pathogen in isolation, but together integration of different kind of data, or together how they are behaving, what is their response, that kind of thing also, because the ethnicity across the several countries will vary, the pathogen strain also varies, so their interaction might be different in depending on the strain or the genetics of the host. So that can be also looked into.
So I'd like to add quickly. Oh, please go first.
So maybe just to add on one half point about different species, different strains, you find that someone may actually think that what they have found is really novel, and yet in actual sense someone out there has already found this pathogen, and maybe they haven't yet published that actually this pathogen is there in the population. So maybe really publicizing what we do would go a long way. Thank you very much.
Yeah, I have a very general question about this flagship project. What's the really outstanding questions? And in this flagship project, what do we want to answer? Do we want to answer, or do we want to identify the pathogenicity of the unknown diseases? Or do we want to make bigger conclusions by integrating many countries?
Thank you for nice and critical questions. We have been taught to lead this session, so actually, but I think the goal here today is, it is also because of the HCA Asia network, to show some diversity across Asia. So how the infection status, the impact is different across the Asian populations is main goal. So why they're different and why not why they're sometimes different but not different. And yes, this is, and of course, but just showing results is not, and of course we can find much more biological aspects underlying these differences. This will be fine. But I think maybe the primary question or motivation is how to set the diversity across infection point. Of course, making target identification somehow is more higher levels of the goal, but in some sense, such kind of study can be done by single institute, right? So we want to do what we cannot do with single institute. So this is one goal. Yep. Oh, yep. Jay.
Yeah, I agree. I think we need to come up with a question at hand. And I think if the question is ethnic diversity, then perhaps it's better to consider EQTL, so what AIDA is doing, but stimulated. Right? So you have a controlled set of compounds or molecules of virus that can be tested across all labs. But the variation comes from ethnicity, right, or the source. So you could consider stimulated EQTL for AIDA. So AIDA 3.0 is something that could be considered for this group, perhaps, because if you do a pathogenic variation plus ethnic variation, you're creating a lot more complication. So it would be very hard to tease apart what is causal and the effect. So something in the line of how can we create a good experimental design that can really address the ethnicity. Second thing I think this group can do is tools and access, and say a country has case of infection or there's an endemic. Is there a way that can deposit their data and quickly evaluate what is the situation with those individuals or in that community? Today, earlier we saw Gmail detecting some viruses. Is there a rapid way of evaluating the immune state, especially in developing areas, so that they can really benefit from the wealth of data that this community has generated? So hopefully, both experimental design, infrastructure from data could also be very useful.
Thank you. I fully agree with your suggestion. Thank you. Oh, yep, please go ahead.
I was wondering if we're thinking about single-cell genomic technique to do this, this flagship project, because once we think about single cells, the multiplicity of infection will change for different cells.
Yes. Yeah, actually, because your point is very important. So first, because we need to be successful in terms of science, we should definitely try to get the new technology. So maybe discussion is just 5' single cell RNA-seq is fine, or we should add more additional technology. This is one point. But the other discussion point that we have said, maybe because this is sometimes difficult to shift the biospecimens each other, so the protocol should be somehow feasible in at least multiple countries within these groups. So the technology only feasible in one institute is sometimes difficult to be applied. So, but I ask everyone for which kind of technology, for example, here. So this is the list of the technology which we have discussed this very short time. So which kind of technology we should definitely apply this? So I think, of course, TANS droplet is one of the core technology, but yeah, single cell RNA-seq or multiomes stuff. For example, maybe see some response to the infection or vaccination. Maybe people recommend not only RNA-seq but also ATAC-seq because they are more dynamically reflecting the changes. Yeah. So
I can add a little, or maybe we can aim at like a technology and we use that as a goal, right? So maybe like countries who don't have technology yet, maybe you could use this as an opportunity to enhance the capabilities in like developing countries. So like we could use Thailand as an example. So we start using single cell technology in context of infectious disease. So we got help from Sarah attachment in the UK, and like they helped us establishing the facility, training people, and then like helping us to the point that we can like run our own project within the country. So I think this could be like like a model as well, that like once we kind of getting feedback on like what should we aim for, we could use that as an opportunity to like enhance local capabilities. That would deal with the limitation of sample transfer, and then like all the like data transfer limitation, as well as in terms of like the balancing contribution, right? Because I survey a lot of microbiologists in Thailand, and a lot of them are kind of protective of their data because they feel like like if they are too open, maybe in the end they would just end up being like sample contributors. So I think this is an area that like we could talk, we could discuss on like what should be the path forward, right?
Maybe let's simplify the discussion point. The first is yes, which technology is best regardless of viability, feasibility. So this is the best in terms of science. The other is how we should make feasibility across the country. So let's first talk of this. What is the best things regardless of feasibility? So, for example, which technology we should definitely apply, especially the biological aspect of infectious diseases. So, yeah, do you have any kind? So, for example, we like this technology because of this infection stuff.
So I'm obviously very biased to 5 points. But I want to say the best technology could also be the cheapest technology.
Cheapest.
Because I think the ultimate goal, of course, science and genetics and variation, all fun and exciting. But ultimately, for this infectious disease, is how informative is this to the society? And the people who are infected and clinicians who are seeing these patients, how quickly can they diagnose, stratify, provide the right care? So I think that should be, I know there's a human cell atlas, so we should be talking about healthy atlasing, but ultimately what infectious disease should aim for, I believe, is point of care or clinical patient care and society meeting that needs. So it has to be cheap and scalable and provide the right platform across the region, not just in one country, that can leverage the resource that we generate. So what could that be? The cheapest and most scalable approach?
That's the cheapest. Maybe nanopore rather than PacBio.
I still think, I agree, in my opinion, right, 5 round with BDJ, but then multiplexing, as many sample as practically possible. So maybe that's still viable. But I can hear others as well.
So, I mean, did I hear it right? Was it cheap-based technology or cheapest technology? Inexpensive technology. Okay. So, now my comment. So my comment may sound a bit it may make sense or may not, but I can't stop commenting. Hello? When the perspective comes clinical application, since I am posted right now in a clinical ambience, the problem that's faced, that is most of the time I get questioned that what you do, what are the clinical implications of the same, why only you study, what are the clinical applications, and also, I mean, even these are the questions from reviewers as well, because from transcriptomic angle, I got these questions as well. So, as we have seen, there is inclusion and exclusion criteria very stringent for any clinical sample inclusion and exclusion. So in this case, for this atlas, not only for healthy samples, but also for sample dispatch, for any disease samples also, I think the inclusion and exclusion criteria should be very, very stringent. This is one part, because, for example, for chimerism, we can't think about, there can be 2 different or 3 different types of cells in a particular person. So in that case, this single cell transcriptomic, I don't know, this can become questionable. I don't know, I think so. Now coming to my main question, that is, as we know, when the thing is statistical analysis, when statistical analysis changes even a bit, the entire analytic part gets changed. So when the data sharing comes, so when I'm sitting on one part of the globe and I'm doing an analysis and I am sharing the data, then another person is sitting on another part of the globe and using my data and interpreting it. So there should be I feel an international scale, like it happens with clinical data. Like we know for BCR ABL, there is an international scale which will not ask any question that what data you are saying is originating from your country is deviating actually from others. Because if I say that this gene is actually deregulated, what should be the criterion? There should be a specific criterion for me to say, yes, it's actually deregulated. It can be because of some of my platform that I have used or some of my statistics that I have used is showing actually a deregulation. If I would have used a different statistics that would have not shown that level of deregulation, a person who is sitting on another part of the globe have used another statistics would have shown much greater a deregulation and reported that. So I feel for such data sharing, there should be an international scale to show that yes, you can say that now this is a gene, this is deregulated, and now you can report it to an international atlas. I feel so. Because since it's a sharing part.
Yes, thank you. Maybe as long as I have heard correctly, you mentioned the 2 points. The first is the inclusion and exclusion criteria with the patient and sample. I think this is the very important point. I think because each of the target diseases, we should clearly define what is the definition of cases. Maybe not self-reported, but this disease, this test results, and yes, we should definitely clarify this point for this in any kind of diseases. And I think of the latter part of your comments on the data sharing. So actually, yes, we discussed data sharing, but didn't discuss how we should use the shared data. So I think that's the overall scheme of the data sharing and analysis may follow what we have done in Human Cell Atlas and also Human Cell Atlas agent, something like the consortium. So actually everyone, actually the people who joined the analytic group is actually people who provide data sharing. So I think everyone in the community have contributions from data generation and sharing. And okay, here's the data of each disease or somehow. And each of the point we prepare some main flagship paper which describe the overall figure, the result of the data. And this is, and actually who do this analysis, who this part is along with the continuous bi-weekly discussion meeting which we have been having the last 2 or 3 years. And everyone, but also we found that there should be some specific other topics who do the, so I want to do the analysis of this data or this phenomenon or somehow. And here comes the satellite analysis. And people propose what they want to do. And actually, most of the case communities say yes, as long as if there's no super batching with the identical proposal. And yes, the point is everyone proposed the data analytic project, and what they have proposed and what they have obtained is under sharing within the community. So actually, we are strictly following the no surprising rule. And of course, people who can help each other can have to enhance each analysis. And finally, we wanna hope more and more projects coming to be the manuscript. But anyway, I think after data sharing, how we should proceed unless we have good experience as this in consortium. So I think there's not so many problems, but more productive way to project. Is it okay for what I responding to your comment? Or yeah, and
I'm still a bit confused about the final outcome that I was questioning. Like, suppose internationally the data is shared, and it's everyone like suggested that yes, it's worth sharing and it passed the criterion and everything. Now the thing is like that particular sample set which was used for data generation, that's exhausted. I'm talking from the clinical perspective. Now when a fresh sample set will be used, now that will completely give a different, my question was from that angle, that will give a completely different representation of a genetic deregulation. But what should we know? That is the question, but I think there should have been an international standard, like we see for the case of certain cancers, for certain mutation detections, there is an international standard that doesn't make any more interpretation questionable. So now when I compare my data with the already established or obtained data from the atlas, when I'll compare the same, and if I find that yes, it's in accordance, now I'll have to say yes, truly it's in accordance when I can actually compare it or I can compare it with the international standard, because or else just I lose my own statistics maybe and I get a particular value and I'll say it's deregulated, but maybe the value is obviously that will come different from what is already presented in the atlas, because my sample set is different. And even when I use a different statistics completely, or whenever even I do an analysis another day, or even if I do an analysis suppose few hours later with a different sample, it will come completely different, So when I get a value, and now if I suppose I say that yes, it's in accordance with the data that's there in the atlas, or even a part of my data is in accordance with the atlas, I got certain genes which are showing deregulation as that has been reported in the atlas. So I have to actually confirm, but that will be more authentic if I have an international standard for that. I'll have to then compare with a set of standard which is given in the atlas. I think there should be more inclusion from the statistical point of view in the atlas.
Yeah, okay, Vijay.
So, on behalf of, so I've been at Human Cell Atlas, OC, and this is a very, very important question, and it's not solved yet, right? So I think how to create a globally recognized and standardized evaluation platform, so that when we put in a disease sample, that we actually know how deviation that we're observing from the normal set. That is a very tall order, but it is a very mission of the Human Cell Atlas. There are examples and pockets of publications and efforts being done by various groups using AIs and using various statistics to integrate, but there's yet to have a globally recognized international standard to this point. Only thing that we're trying to do right now is at least have a cell annotation, right? That's like the most foundational basic thing. Exactly. So we'll get there. So we're starting with the cell annotation now, with the CAP platform that you saw today, by community-based annotation of what cell type is, and then we'll move on to such efforts to integrate and to create internationally recognized standards for evaluating and diagnosing, which is very hard, but that's the vision.
Thank you, Jay. Oh, yep. Please go ahead.
I just wanted to follow up on 2 points to continue the discussion from earlier. So I think one of the things was, one of the questions that was raised was standardizing the technology across sites. I wonder at this stage whether the more important thing is to standardize the sample collection procedure. And either one of our experiences is that the biggest batch effect actually comes from variation in sample collection. And then because I'm assuming you're collecting stuff like nasal swabs, PBMCs, so these are, you're able to bank more of these. So maybe one viable approach is, collect multiple, maybe 3 samples of the nasal swabs, 3 samples of blood. Whatever teams want to do with the first one, they can go ahead a bit, have it bank, and then people can go back and trace and then rerun those with a standardized set of assays. So maybe that's the easiest thing to standardize, and it will also help to control or limit the number of batch effects across different countries. I think the second point actually is tied to this. So what's the value proposition of what we're trying to do over here? Apologies if this has been brought up because I was in the cancer cell program earlier, but one of the things that I think the world's very concerned about is the next COVID. So pandemic surveillance and pandemic preparedness. And this reminds me of what people are doing in places like Seattle, with the Seattle flu study, where people are doing pandemic surveillance, surveillance of respiratory viruses. So can we hop onto this and maybe maximize the value from existing surveillance programs? So tying with, respiratory surveillance of respiratory viruses, do single cell genomics pad of this, and then just enhance the value to society. Because this is going to be fresh in the mind. I think selling this is going to be an easier proposition for the public. And it's going to make a meaningful impact on public health. Thanks.
Yeah, I'm a computational biologist, and like we haven't discussed most of the competition part. So I feel like this was lacking in the discussion. So in this, there are actually next flow pipelines in the front to analyze what ma'am has discussed, right? The statistical analysis and all. It is basically 100% reproducible. So before starting any of these such large projects, if we make a standardized pipeline, which is next flow based, and then everybody accepts that, and you can also use it in Amazon Web Servers or some it's basically some cloud platforms where you can restrict your data to locate in your country itself and then generate data, and you can upload into once if the parameters and everything are standardized from some point to you can find you can upload your data and get your results, and if for example if two people from two different countries have generated the data, and if both of their bioinformatics pipelines are 100% same, and then you can trust each other results. So this was actually partially solved, and the only thing what we have to do is basically we have to finalize the parameters and you can plug and play. And also there is this GitHub repositories where any person can contribute. So for that you can keep make some repositories and all, so that make sure that all the parameters whatever there is no constraint, it should be open so that any person from any country can see, okay, in your pipeline there is some mistake at specific point of at a line of code. They can change it out, they can suggest it. So these things should be it should be improved.
Yeah, your comments are very informative. Thank you. Yeah, great.
Can I add another point? Maybe just it's my question as well, and my thinking about standardization and everything, right? So of course we all want standard, right? I want just having my data, and then some magic tool would just define annotate cell immediately, right? So that would be cool. But in reality, right, there are new tools coming every day, and newer tools some are better than others. So there's a kind of a balance, right, between what should we do to have a common consensus or pipeline, so that we can all do things in the same way for better control, right, controlling for confounding factors. But at the same time, we should be able to make this flexible enough to accommodate future improvement. So I kind of feel squeamish about the word standards, standardized, right? Because I feel if we're too fixed on this, then we wouldn't be able to adapt when better approaches with experiments why, or the in terms of computational tools why, would be adapt to or improve the current pipeline. So maybe asking for you guys' opinions as well, that what should we do with this, right? I'm trying to be careful, framework, common pipeline, consensus pipeline, versus standardized. So how should we deal with this or balance these two areas?
Sorry, I'm talking a lot, but I think also for this group, it's very important that we engage with the immune network. So within the Human Cell Atlas, one of the 18 biological networks, the immune atlas, they are essentially creating the under blueprint or the reference data set, so that this community can then add on top of that. So what platforms do they use? What are the parameters? What are the next flow pipelines are they using in order that we can readily implement and adopt to that atlas? So I think that's something, a lot of the work has been done for the last 5 years, so we don't have to reinvent it. So let's leverage that and work with their community, and also learn from them on how we can adopt, also improve. And I think the second thing is, Kyung mentioned, sample acquisition. How you get sample is very, very important, and especially for infectious disease samples. healthy donors, the immune cells is okay.
I agree.
You just ask someone in your lab colleagues or something. But in infectious disease, it's very difficult and challenging and needs to be well maintained and metadata has to be health records and all that.
Right.
So that's something this community can also contribute to further fortify the existing immune network.
Yeah.
I just want to make a comment that's probably very general, but there are specificities here as well, in the sense that after all, we want to get to robust inferences from whatever data we collect and generate. What we need to do, it's going to be a long haul. What we need to do is to identify the variables which will impact on the final inferences. So, for example, one of the variables that has been appreciated and there are efforts going on to correct for that, this is batch effects. So batch effects, we didn't pay any attention to batch effects when we were doing genomics. But right now when we do transcriptomics, batch effects are very, very important. So it's critical to identify those parameters or those variables that will impact on the final outcome of the final inferences from the data analysis. And these variables will not be the same across infectious disease, chronic disease, and microbiome. There'll be separate sets of, well, there'll be some overlap, but there will also be some specific sets of variables that one needs to control. I think it's critical to identify those, because if you identify those, then the protocols and the tools can be improved. Unless we identify those, it's going to be very hard to see which direction to go in order to improve the robustness of the inferences. Thank you.
Yes, thank you, Professor. Yeah, I fully agree to you. Yeah, okay, sorry, just a comment. I'm sorry, but we only have 5 minutes left for discussion. So starting wrap up. So first of all, if you have any further comments or opinion, just grab me and Waradon and input your thoughts. Hopefully better than before we get drunk. But anyway, we would like to reflect your opinions on the comment. And actually, we have some good feedback from now. For example, point 1, I heard so many votes for TB and also dengue. Namugun, Namugun, are you hearing? Can you put some additional votes or you're pushing for NTM if you're online? So why
Yes. Yeah. Thank you.
Important?
Thank you for the discussion. I thought the sound is okay.
Yeah, good.
Okay. Okay. Yes, I totally agree with today's wonderful discussion. And NTM, a little bit not uncommon disease in Asia, but in Japan or in Korea or Taiwan or China, the number of patients of NTM is very increasing. And also this is very interesting because the whole genome sequencing bacteria and also host genetics of NTM is now we are doing a meta-analysis. It's a very good model for international collaboration. But I also think that for example, influenza is very important for next pandemic. So I think that NTM is a very good model for future further international collaboration. Thank you.
Okay, thank you. Okay, so maybe if you have any further inputs for this, let us know. And the second point, select target tissue, what I heard from this session is mainly blood and also swab is also. I think blood and swab is the people most easily get from each country. So these two are the major point. And technical method, I think feasible and cheapest is important. So single cell RNA-seq fibrin is fine. But if you can put some additional bolt, maybe if the additional institute which can do more, we can do more of non-cheapest option for each institute. So it's ATAC-seq or long reader, we can put 0.3. And 0.4, this is important. So I think we do not restrict only one disease, but for each of the candidates, we should definitely define what is the inclusion and exclusion criteria, and what should be the strain level information we should get from each patient. We should clearly define different. And 5.1 to host pathogen, I forgot to say is, yes, we should definitely ask you to get host genetics as well as transcriptome and pathogen. So actually, what we learned in COVID-19 pandemic, some people collect pathogen but not get genomes. Some people get genomes but do not pathogen. So actually, the power, we definitely make the scheme to collect all of this information to get host pathogen because, host genome is most diverse things within Asian countries. And so grant proposal got found. This is, of course, we should apply to C0 and things. But anyway, what we learned from this session is we should make nice pipeline in terms of IRB or somehow how we can get and also the protocol, how we do collect the sample or how we do stimulate the sample or how we do the analysis, maybe the shared protocol which everyone can do, and also make some IRB network so easily to share each other, something like IDA. And we should definitely put this scheme, it can be applicable to the upcoming new disease. So once the new disease, infection disease comes, this network can quickly assess and coordinate. So this is the important one. So this is the brief input from what I learned from this. But yes, Brad, do you have any comments? Thank you. Yeah, but so Anderson, this time over. It's now. Time is over now. Okay, so maybe I first imagined that the round one hour was too long for us to discuss, and I was really afraid everyone gets silence. But there are so many hot discussions from the audience, so it's really happy to. So is there anyone who can say, I want to say last minutes for this, please raise your hand, and any comment is welcome. Is this anyone fine? Have you said anything you want to say? Or you can tell both afterwards. Yeah, any comments is fine. But please notice the networking is the most important purpose of this meeting. So this is the reason why I ask you to activate you to say or introduce yourself. Please communicate in each other in this room because it's a nice community for the one purpose. I hope you have some enjoy time for our dinner somehow because there's so many times to communicate. So I hope you will enjoy this meeting for a variable time for us. Okay, maybe we should wrap up the session now. Thank you for everyone to joining this wonderful session and active discussions. And thank you for everyone who joined by the Zoom. I hope to see you in discussion in person next time. Okay, thank you everyone. Maybe we need to show some summary of this discussion tomorrow, right? So we are going to actually short slide for this. So as you can maybe ask us to reflect your own opinion to this. Of course, and also we need to keep working on networking. Yeah, but yes. Someone taking minutes from the meeting, so. Oh yeah.